It is with great pleasure that I announce that the E. Donnall Thomas Award was bestowed to Dr. Carl H. June, and he will be giving the E. Donnall Thomas Lecture entitled New Cars and Used Cars for Leukemia and Beyond. Dr. Carl June is the Director of Translational Research at the Abramson Cancer Research Center at the University of Pennsylvania and an investigator at the Abramson Family Cancer Research Institute and Professor of Pathology and Laboratory Medicine at Penn. He is a graduate of the Naval Academy in Annapolis and Baylor College of Medicine in Houston. He has graduate training in immunology and malaria with Dr. Paul-Henri Lambert at the World Health Organization. Afterwards, he did his postdoctoral training in transplantation biology with Dr. E. Donnall Thomas at the Fred Hutchinson Cancer Research Center in Seattle, and then went on to found the Immune Cell Biology Program and was the head of the Department of Immunology at the Naval Medical Research Institute. In 1999, he moved to Penn, where under his leadership, the translational research team at Penn has accelerated research advances in the development of therapies that use the immune system to fight cancer. He and others have been famous for pioneering the development of chimeric antigen receptor T cells, or the so-called serial killer T cells. His team has also been pioneers of other novel immune-based therapies using both vaccines and cells. The promise of these therapies are in our imagination, that the day that we can cure patients simply using their own cells or their own immune system by engineering them. Please join me in congratulating Dr. Carl June for this well-deserved award.
Thank you very much. Thank you. Well, thank you very much, Sergio. It's an amazing honor to be here to do this, and also to Paul Martin and the selection committee. So I'll talk about CAR T cells, and the first thing I have to say is this is, I think, going to be something that bone marrow transplantation is best positioned to take over as a new field. Disclosure I have is that my research and clinical trials are now funded by Novartis. And I want to take some time to thank the people that helped me get where I am here, and some of them are shown here. So I had an amazing fellowship in Seattle from 1983 to 86. And up at the top left, you can see Don and the team there with Fred Applebaum. I learned to catch salmon there in Seattle by Larry Lum. And I was in John Hansen's and Paul Martin's lab in immunogenetics for several years there. Learned first about adoptive therapy from Mac Cheever. And we had an amazing fellowship group. I've put some of them there with Claudio Anicetti, Ollie Press, and Craig Thompson. It was just an amazing time there, and we all taught each other a lot. So when you look at cancer, the cancer biologists have said there are 6 hallmarks to make a cell be able to become cancerous. And it was only recently that they added a 7th hallmark, which is that the immune system must become tolerant to the cancer. And that's been a major leap forward for the field of cancer. Bone marrow transplanters have known for a long time that the immune system has an effect on cancer, but it has taken a long time to take and make that front and center with cancer biology. So there are a number of ways now to overcome tolerance, which the cancer cell must impart on our immune system in order not to have it rejected. We've started with therapeutic vaccines, and then most recently the development of checkpoint blockade with antagonists to CTL4, and PD-1 has been done to unleash some of the tolerance mechanisms that occur in the tumor microenvironment. But what I'll focus on are these adoptive cell therapy approaches using T-cell clones, or TILs, first in metastatic melanoma. This will be commercialized by Lion Biotechnologies. They're undertaking randomized trials, and Yvonne Borello is here, who's using a similar approach, the marrow-infiltrating lymphocytes in myeloma. So these are natural T cells. The other 2 forms are genetically modified cells, in one, the so-called CAR approach that I'll describe, which a number of companies are now attempting to take into late-stage trials, and then T-cell receptor engineered cells where new specificities are introduced. So we'll be at a point where we have new tools in our armamentarium for cancer therapy. We now have, in addition to the standards of chemotherapy and radiation, we'll have cell-based therapies that will be FDA approved probably as soon as 2016 based on current projections by several companies. And one thing that you can say is that the chemotherapy in general targets the tumor. Immunotherapy in general is directed at the host immune system, so it's more device independent. And by amplifying immune effects then is the goal. So there are some considerations when we look at how we may have an immune-based therapy. If you have patients with late-stage lymphoma or leukemia, they oftentimes have more than 1 kilogram of tumor, which would be about 1,000 billion cells. T cells kill on a 1-to-1 basis, and in general, you need to have an effector to target ratio close to 1. So you need a lot of T cells if you have a lot of tumor cells. So many previous immunotherapies failed because the effector to target ratio in vivo never came close to 1. And so there are two ways with adoptive therapy to try to achieve that, one being to infuse very large numbers of cells, which has been done with the so-called TIL therapy, where metastatic lesions in melanoma are resected and expanded to large numbers in vitro and then given back to patients. The approach with CAR T cells is one where to reprogram the cells so that it can then divide massively in patients, and that's what I'll focus on. So CAR T cells are synthetic molecules. They don't exist in nature. The first one was a so-called first-generation CAR made by Art Weiss at UCSF, and what he did was fuse the CD4 extracellular domain of the CD4 molecule to a molecule in the T-cell receptor called the zeta chain. And that was used to study the signaling of the T-cell receptor. But it was later realized that this could retarget cells towards HIV because CD4 binds HIV envelope with very high affinity. And in fact, cells with this CAR will kill HIV-infected cells as well as a CTL will. And then over the years, new design improvements have been done to these CAR cells with Zellgesar grafting antibodies on to the zeta chain rather than a natural molecule as had been done here earlier. And so then you could retarget a cell to virtually any target that you can make an antibody to. And then the field over the last decade or so has worked on making more complex signaling domains so that when the CAR cell binds its tumor target with this antibody, it will have signaling through here that will both trigger killing of the tumor cell as well as proliferation. And the ones that we have taken into the clinic have a molecule called 4-1BB as a co-stimulatory molecule in addition to Zeta, and other groups are using CD28. And I'll show a little data where we've now tested ICOS, which is a molecule related to CD28, and as a gene duplication as an intracellular signaling molecule. And this work has been done by Sonia Gaydon, who recently left my lab where she was a postdoc. So in this case, she's taken a CAR that binds mesothelin, and we're testing for pancreatic cancer, and tested varying signaling domains, including CD28 or Zeta only, and now ICOS. And what she's shown in a series of experiments is that this will program the cell to become a Th17 cell if it has ICOS. And Th17 cells are the cells that cause autoimmune disease. And so they're very good at breaking tolerance. And our data in animal models shows that these work better against varying tumors. And this is one example where she put different signaling domains in different cells, so in either the CD4 cells or the CD8 cells. So this is the CAR that we have in clinical trials that has 4-1BB and a Zeta signaling domain. And these are xenografted mice, so-called NSG mice, that have pancreatic cancers in the flank. So this untreated mice die after about 6 weeks. And if we use the one that we're using clinically, or the 4-1BB and zeta, it causes stable disease. And if she puts one that has ICOS, 4-1BB, and zeta, she's able to eradicate these pancreatic cancer cells via the anti-mesothelin CAR. So that works better, and we think the reason is that there are more cells that proliferate in the patient. So these, in the mouse, these are the number of T cells per microliter of blood in these immunodeficient mice, and whether we look at the CD4 CAR T cells or the CD8, it's having ICOS is what promotes better proliferation in these mice. And that, as I'll show you in our human trials, what correlates best with regression of leukemia is proliferation of the CARs in the patients. So we are going to be testing these later. This just shows that there's differential cytokine profiles. If you have an ICOS-based CAR, which is this, there's much more IL-17 produced when it sees a target compared to these other CARs that have either 4-1BB or CD28. They're about all equivalent when it comes to whether the tumor surrogate target induces interferon gamma, but only CD28 causes IL-4, so that's an IL-5 secretion, Th2 cytokines. And we find that the CD28 also promotes more IL-10, which is immunosuppressive, and in contrast, TNF is made very well by all three CARs, but most by the CD28. So the nature of the signaling domain can change both the proliferative capacity of the CAR and the cytokine secretion pattern. And in addition, it depends on what subset of cells that are used, either CD4 cells or CD8 cells. So at this point, we use bulk T cells and put the same signaling domain in both the CD4 and CD8 cells. In the future, we'll probably be separating CD4s and CD8s and putting ideal signaling domains in each subset. So this just says that the different CAR signaling domain can program how the cell works later, and I think for different kinds of cancer, this will be important. So over the years, we first began our first CAR trial in the late 1990s in patients with HIV with that CD4 zeta CAR, and now, as of December of last year, we treated 236 patients now with either cancer or HIV with various forms of engineered T cells. And we've never had an adverse event where due to the genetic engineering in the T cells. So we've never had a single case of transformation of the T cells and have more than 1,000 patient years of safety. So engineering T cells in this way appears to be much safer than giving patients cytotoxic chemotherapy as far as looking at secondary cancers. So every patient on a gene therapy trial in the United States is required to be followed for 15 years. So we're getting quite a safety database now. So the initial trials did start in HIV, and then in the early 2000s, and CARs were then tested in cancer patients. And the first generation CARs that just had the zeta chain only were tested in a number of cancers, kidney cancer, ovarian cancer, and neuroblastoma. And the CARs didn't have any anti-tumor effects. They were safe, but they didn't have prolonged engraftment as we had seen in HIV. And it was these disappointing results were most likely due to poor engraftment and proliferation of the CARs in the cancer patients with first-generation CARs. So then the field developed a number of second-generation CARs, either based using CD28 or 4-1BB. And with the FDA, it was decided that these more potent CARs would be tested in patients with B-cell malignancies. And therefore the targets were either CD19 or CD20, the rituximab target. And that's because it was known that CD19 and CD20 are not on the hematopoietic stem cell. And so these CARs would not cause aplastic anemia as a side effect. And with the knowledge that you can have congenital B-cell aplasia and you're fine as long as you get gamma globulin replacement therapy. So the prediction would be if these CARs are successful, they will induce B-cell aplasia, and that's a measure of whether the CARs remain functional in patients. In general, myeloma is said not to have CD19 and therefore is not considered to be a target, but I'll, at the end, show some data where this appears perhaps not to be true. So our initial trial was started by David Porter in 2010 with the second generation CARs, and it was in patients with late stage CLL. And so the patients donate their own cells via apheresis. The T cells are manufactured over a 5 to 10 day time with the introduction using a lentiviral vector to put the CAR in their T cells, and the T cells are cryopreserved at the end and then given to the patient via simple infusion, just as in a stem cell. They're frozen in DMSO, thawed at the bedside, and infused. And so our first patients were reported in 2011, and all three had very advanced chronic lymphocytic leukemia with up to 7.5 pounds of tumor as assessed by imaging and marrow examinations. And what we could show, because you can track these CAR cells in the patients either by flow cytometry or by PCR, is that each CAR cell was a so-called serial killer cell that could result in the death of more than 1,000 tumor cells. And that's either the CAR cell itself repeatedly killing other targets, or its progeny, because the CAR cells have proliferation when they encounter tumor targets. So this is the first real examples of a living drug. These CAR cells remain in these patients now more than 4 years later. And so the remissions have been durable, and the patients continue to have B-cell aplasia, and they're basically having the benefits of a targeted antibody therapy, in this case anti-CD19, but they don't have to have the recursive infusions. The T cells synthetically make the antibodies, and then they have CD19 specific killing. So that trial was finished by David Porter. The first 3 patients were, initially reported, and then the others. And the overall response rate in these patients with advanced refractory CLL was 57%, and no patient who's achieved a CR has relapsed. And so far, those effects have been durable. And what we have found as a biomarker of response in these patients has been whether or not the T cells, the CAR T cells, proliferate in the patients. And as I mentioned, we can track these cells genetically in the patients by PCR, and this is quantifying the number of these CAR T cells on a semi-log plot in copies per microgram of genomic DNA, and we've done this either in blood or bone marrow. And these are the first 10 CR patients that we had. And when you look at these and do a back calculation, if you have 10 to the 4th copies per microgram of DNA, that's about 6% of the cells are CAR cells in the blood. And all the patients who have complete responses go above that line. And the patients who have not responded have engraftment of the CAR cells, but they don't proliferate in the patients for various reasons that we're studying. So the CAR cells need to proliferate in the patients, and then they persist in about 75%. The second trial we started then was in acute lymphoblastic leukemia, pre-B-cell ALL, both in children and adults. And as it happened, our first protocol, we had a separate protocol for the pediatrics, for the adults. The first protocol to get open was in the pediatric ALL. In adults who have ALL, bone marrow transplantation has not been nearly as successful as in myeloid leukemias, and if you have relapsed ALL, the survival at 2 years is about less than 10% in adults, so not the same kind of outcome as we have in children. But in children with relapsed leukemia, the outcome is also poor. Our first patient was Emily Whitehead, treated in April of 2012 by Stephen Grupp, who is her physician, and she's now disease-free and more than 2.5 years later, and has become an icon in cancer. She was featured on the front page of the New York Times when we reported her case in ASH at 2012, and then 2 weeks ago she was with President Obama, and when he and Francis Collins announced the initiative, the Precision Medicine Initiative, and it was quite astonishing. Emily, who is about 4.5 feet tall, was sitting next to Kareem Abdul-Jabbar, who is 7 feet 1. And the reason Kareem Jabbar was there was because he has CML, and he's been on imatinib for many years. And So that we have now two kinds of targeted therapy for leukemia. We have drugs like imatinib, and then we have retargeted T cells. So this is Emily's case. She had a very high-risk ALL because it had a Ikaros deletion, and she was just chemotherapy refractory from the get-go. And when she was treated finally in April of 2012, there was no chemotherapy that was given to her. She had high-dose cytoxan and clovarabine 6 weeks before and had had persistent leukemia. At the time of CAR infusion, she had 90% blasts in bone marrow and numerous visceral lesions. And so the CAR T cells were infused with no other chemotherapy. And because we can clonally track, and this gets to the kind of the issue that Bob Gale was talking about, we can look for MRD in this disease by measuring the IgH clonal signature of the leukemic cells. So at baseline, in the bone marrow, when we looked at 317,000 cell equivalents, 99.97% had one IgH rearrangement. That was her malignant clone. And then she had a tumor lysis syndrome and a very severe cytokine release syndrome 5 to 8 days after we treated her. And on day 23, she had a bone marrow examination, and now there are many fewer cells in her marrow, and it's down to 89% of them were malignant. And that you can see these clearance kinetics. At 180 days, she had a bone marrow, and now when we look at 1 million cell equivalents, there are no detectable malignant clones. So she remains tumor-free now 2.5 years later. She has an ongoing B-cell aplasia, but in this case, we can show that there is at least a 5 log tumor reduction in this patient, solely due to the administration of CARs because nothing else was given. It did take 6 months for the tumor to become immeasurable using sensitive assays. We now, as of ASH, landmarked at late November, had 135 patients treated with these CD19 CARs, including 41 pediatric cases. And of these, 28 had already relapsed after an allo transplant. And 35 out of the 39 children have had complete remissions, a 90% complete remission rate. So it's quite astonishing. Emily being the first, but we see a very high and reproducible rate in both children and adults with ALL. And this is the survival curves and duration of responses in the first 51 patients, of which 39 are pediatric and 12 are adult. So there's some fall off, but after 6 months, it's 8 months, we haven't had relapses, again, with Emily being the one that's farthest out. And this is the kinetics of clearance of these CARs in the first 2 patients we treated, either with CLL or ALL. And these are different times. This goes out to 4 years, and this is out to 2.5 years on this axis. But this is a semi-log plot of the number of CARs in blood. This patient had a tumor lysis syndrome 23 days after he was treated for CLL, and he still has, you can see this decay rate, the lower limit of detection of the CARs in blood, which is in blue, or in red, which is in bone marrow. And so he remains lymphopenic and B-cell aplasia, but with no CLL. In contrast, the clearance of the CARs has been more rapid in Emily. She, 6 months after infusion of the CARs, had thymic reconstitution and a large outburst of new T cells, and has a normal immune system as far as everything that we can tell except for B-cell aplasia. But here now at 2.5 years out, she's right at the limit of detection, and she's beginning to get B-cell reconstitution as judged by hematogones on bone marrow aspirates, although she still has, and then we can't detect leukemia. So there have been toxicities from CARs, but to date they've all been on target. We've had B-cell aplasia, cytokine release syndrome, which is, as I'll show, related to tumor burden, and is now treated with anti-IL-6. We initially treated it with high-dose corticosteroids, but we now treat with cytokine therapy using anti-IL-6 receptor, which neutralizes IL-6 signaling. And a subset of the patients get macrophage activation syndrome, and as with blinatumomab, they can have neurotoxicity that's reversible. And this shows that the cytokine release syndrome is related to disease burden. So if they have a packed marrow, say 80 or 90% blast, then they have a high chance of having grade 4 cytokine release syndrome. And if they have low marrow burden at the time of treatment, then there's much less chance of having severe cytokine release syndrome. So we've just recently begun a pilot trial with the CD19 CARs in myeloma, and this is led by Al Garfeld, Marcella Maus, and Ed Statmauer at Penn. And as you all know, myeloma is traditionally classified as being CD19 negative. But there's data that myeloma begins from a clonotypic B cell that is CD19 positive, and then people go through the MGUS stage to where they have advanced myeloma and many additional genetic lesions. And there's now emerging data, there's some plasticity in this. And the cells, as Bob Gale showed the Peter Quisenberry figure, stem cells may change, tumor cells may, depending on their microenvironment, have different characteristics. And it's reported that at a low frequency, you can find the clonotypic CD19 positive cells in the blood of myeloma patients. So we have begun this trial, either saying there may be a CD19 positive precursor, and by targeting that with CARs, we might have an effect eventually on the myeloma more mature cells. And it's also possible, we have data that I won't show tonight, that the CAR can target less than 100 molecules on the surface. So much below this, it's much more sensitive than we have, say, as a flow cytometry assay, and there may be low levels of CD19 on myeloma cells. So we've begun a pilot trial to test this, and it's to take patients after an autologous stem cell transplant and give an infusion of the CD19 CARs, and then measure time to progression. And this is for patients who have already relapsed after an initial auto transplant. And the secondary endpoint of this then is to look for a remission inversion. And so it's a pilot study of 10 patients, of which 5 have now been infused. And the primary endpoint is safety and feasibility, asking whether or not we see the same things as we do in B-cell leukemias with B-cell aplasia and CAR engraftment, but also to measure this time to progression. And at this point, as of December, there had been 4 patients enrolled, and the first patient was given, had actually plasma cell leukemia and very advanced myeloma, and was given a dose of 300 million CAR cells. And she had very severe cytokine release syndrome that was reversible, but had a more than 3 log reduction in her tumor mass as assessed by several mechanisms for 4 months. And the second patient then, and we've given a dose reduction based on the cytokine release syndrome. And the second patient I'll show, but this is, the first patient had cytokine release syndrome with IL-6 going up about 3 logs over baseline. These are the CARs in red, and it happened at about day 15 after infusion of the CARs. And this is staining in the blood at baseline, day 9 and day 16. And on day 9 after infusion, she had about 20% of her peripheral blood T cells were CAR cells. And this shows serum ferritin and C-reactive protein. C-reactive protein is an IL-6 driven gene, and it's part of what we see in the cytokine release syndrome. So this is very much what we have seen in CLL and ALL, so that can be triggered in myeloma. Our second patient is shown here, who had a dose reduction in the CARs, and this was basically done as an outpatient. She'd had a melphalan 200 autologous transplant, with an IgA myeloma, and her paraprotein went down a little bit, and then started progressing again after 3 months. And then she got a second transplant, but with dose reduction, with a MEL down to 140, but this time with CARs given after, and she's now has an undetectable paraprotein. That's now out to 6 months. So that she's had a very gratifying response that she didn't get with higher doses of alkylating agents. So the mechanism of this and durability obviously remains to be established, but it looks quite promising. At this point, there are a number of CARs now in development by companies in the US and Japan and China. This is a listing of just some of them. alphabetically. And there are a number of academic institutions now testing CARs against many different kinds of cancers beyond just CD19 and CD20. So it's early days to see how far this will move beyond B-cell malignancies. But every B-cell malignancy that we've treated so far has responded to the CD19 CARs. And I think we'll be facing shortly the ability to combine adoptive therapy with other kinds of therapy, including bone marrow transplants and checkpoint therapy. And there have been studies shown in mice that if you give checkpoint blockade with CARs, that it can have a very potent synergy. So I'm sure we'll be seeing this in the next year or so to see if we can enhance some effects in other tumors. So where we are at this point is the beginning of a new kind of therapy that's a personalized therapy with a patient's own cells. The only precedent really is a bone marrow transplant. And people early days in bone marrow transplantation said it could never become channelized. They said it would be impossible to scale. And as you all know, in 2013, we had the 1 millionth bone marrow transplant was done. So it is possible to scale difficult therapies, but you have to train people and develop new technologies. And so we will go from where we have the pharmaceutical industry and the biotechnology industry making recombinant proteins and medical devices to where we have a fourth pillar that will be cell therapies. And I think this will be implemented by bone marrow transplant physicians. There'll be a number of issues that they know well, which is it's a patient-specific so-called N of 1 issue. So each T-cell therapy that we give is FDA, it has to be FDA approved because it's patient-specific. And bone marrow transplantation never had to go through FDA approval because it was grandfathered into the practice of medicine. There are issues of whether or not the T-cells will be grown in blood bank-like settings or will they be grown in central manufacturing plants. In the U.S., the FDA policy is that these will, if they're genetically modified, will be done in manufacturing plants. So just to summarize, CAR T-cells have a number of potent effects in ALL and CLL. Registration multi-center trials are now underway by Novartis. The cytokine release syndrome is treated not with steroids now, but with tocilizumab and other agents probably that block the IL-1 cytokine pathway. The CARs have also robust activity in lymphomas and maybe in myeloma. The most important aspect now remains actually an engineering issue of learning how to grow cells with automated instrumentation rather than by hand, as we currently do it at the University of Pennsylvania with scientists and technicians. So a number of people that I'd like to thank for this work, both at the University of Pennsylvania, Children's Hospital in Philadelphia, and at Novartis. And I'd like to thank you for your attention.